All right. Okay, do you hear me okay?
Yes, that's fine. Just a little bit of feedback. Maybe get a bit closer to the microphone. Thanks.
All right, okay. So hopefully you hear me now. Good morning, everyone. Well, this study is part of my ongoing PhD research, which is more or less about the needle now. And as the title suggests, we are particularly interested in understanding a little bit better the phosphorus cycle, potentially involved in prebiotic chemistry on the early Earth. It was really good to have Sascha's talk before me. She has paved the way, I guess, in terms of introducing the links between phosphate and carbonate nucleation in general and in the new Proterozoic. So I guess we will move on further back in time now. Okay. So just giving you a little bit of background before with a few points relevant to our study. Well, there are certainly many things important about phosphorus and phosphate, as Sascha just highlighted. Although not everyone likes phosphorus, but that's certainly, well, anyway, it's certainly a very interesting element. And, well, although there are certainly many things that are relevant with respect to phosphorus and phosphate in the environment, as we may know, as you may know, there is certainly one very remarkable thing with respect to phosphorus that is the focus of what I'm talking about here. That despite the fact that it is present in all organisms and that it comprises the backbone of extant biochemistry and biology as well, phosphorus is not abundant and readily available in most rocks, the primary source for this element, of course. So for this reason, how this concentration problem was overcome to provide enough phosphorus to get, like, say, kickstarted has become known as the phosphate problem. So we know, and we heard before about the dolomite problem in the oceans and in the geological record. I guess that science, we really love some good problem, or at least to label things like that. We have now a phosphate problem, and, well, just recently though, it was proposed that alkaline lakes on the early Earth, they may just have been able to provide the necessary high concentrations of phosphate in solution. Moving on, and investigating modern alkaline lakes and experiments simulating these conditions. Toner and Caitlin just recently observed that phosphate accumulates up to several millimoles under high carbonate alkalinity, which you can see here, phosphorus in log scale through the y-axis and dissolved inorganic carbon, representing also a good estimate of alkalinity, and the axis. And you can see here the punch line as well. They measured and collected data from publications as well from several different lakes throughout the world. And we observed this very nice link between high phosphorus content in these conditions. So, well, this still makes us think that we still lack a geologic record suggesting that similar physical chemical processes may have occurred in ancient alkaline lakes. So definitely this begs the questions that we're addressing here in our study and that you can read there. So could then ancient alkaline lakes have really reached high phosphorus concentrations in solution? And if so, can ancient alkaline lake deposits preserve a record of their original solution chemistry? Therefore, the goals of this study are to investigate a potential model from the geologic record whose conditions of deposition and preservation may have been suitable for us to test these hypotheses, and also to further explore the relevance of these observations to the distant past. So now, well, a lot of these approaches that we are using, Sascha has covered already. So, well, this is one of our approaches that we are using to investigate in detail the geochemical characteristics of the rock record. We are using in this empirical part of the study, looking at pulp rock geochemistry, for instance, from samples collected from almost 200 meter continuously poured interval from Santos Basin that I will be talking a little bit more in the slides to come. As well as we are looking at in situ analysis, applying different instruments and resulting in further collaborative work, such as the SSRL shown there. The second approach is based on modeling of the conditions of our carbonate phosphate system, supported by our experimental and empirical observations. I will not dwell here anymore because Sascha has already spoken about this, but this is the other approach we're using, experiments, to further test mechanistically and understand processes that we may observe in the rock record. So turning now to our analog system for the more distant past, these non-skeletal lacustrine pre-salt carbonates of the Barra Valley Formation offshore southeastern Brazil. They have been deposited during the early Cretaceous, during the rifting of Gondwana. As you see here, this map to our left is rough paleogeographic reconstruction, giving us a perspective, the paleogeography context, as well as perspective in scale, although it's not showing the deposits on the West African margin. But we are interested here in this region, in Santos Basin. The map to our right shows a rough structural perspective of this region, and also indicates where our samples are coming from. So I will not talk much about this, as well as because we yesterday had some really good talks about these carbonates from both the Brazilian and African margins, as well as we heard some potential analog studies. Perhaps just to, in some sense, to shift or invert the perspective and propose that carbonates and these systems can be a testing ground for assessing the preservation of environmental conditions that we normally study in, for instance, in modern environments, and we usually extrapolate them to the very distant past. Even though these basins have undergone some interesting deposition or diagenetic history, they have not gone under so much structural deformation, for instance, and weathering and so on. So anyway, to ease your expectations, I jump now straight away to the rock record to answer our initial questions, which you see there, back there on the screen. Here we can see the profile of total phosphorus concentration measured in bulk rock samples with ICP-MS. Here we have mineralogy on the left side of this image, determined via polar X-ray diffraction. And you see the core is essentially composed of these three minerals: calcite in this light color, here light blue, dolomite in this darker shade of blue, and quartz in gray. We also see detailed facies description of nearly 500 samples in 10 sections from study João Paulo Gomes and authors published early on in this year. So here we see in situ samples, like the classic shrubby and spherulites. Okay, I'll move on faster. We can see here that essentially most of the phosphorus is apparently related to this shrubby facies, which is normally regarded as depositional primary sin sedimentary texture, and the phosphate, high phosphate, phosphorus in this case that we see very high. And here is a 9-point running average of the total phosphorus content, is usually related to these fabrics as well that are predominant in this part of the interval. So moving on to another approach, which is this figure here. We show modeled phosphate concentration in solution with varying concentration of calcium. This model is based on mirror equilibrium calculations, assuming an apatite precursor, and the pH values of 8 to 11 shown here. So all these curves are actually these solubility curves of this phosphate precursor. So we can see that under this whole range of high pH values, we can have high phosphate concentration in the solution, provided that calcium remains low in the same solution. Otherwise, we may certainly expect that some insoluble phosphate minerals will start to precipitate. So this slide just summarizes some important observations from a few of our crystal growth and precipitation experiments. As I don't have time to dwell here, the main take-home message is that we observe that increasing phosphate that you see shown here in the solutions, the supersaturation state with respect to calcite can be sustained for longer periods of time. So these experiments were all run in the same conditions, apart from phosphate that varied.
Sorry, so, about 30 seconds before the screen share ends.
Okay, just the summary of our observations. We can see that phosphate concentrations can reach up to at least a few hundred millimoles in high pH. You can see that from our model calculations. You see that we observed remarkable concentrations of phosphate occurring in these Cretaceous lacustrine deposits, suggesting that phosphorus was likely available in solution during the deposition, and that phosphate increases induction time and sustained supersaturation. We see that from the experiments, and these processes could potentially have provided concentration mechanism in ancient alkaline solutions more generally. Okay, so finally, I would like to thank Petrobras for a generous grant and support of fundamental research. I would like to thank my lab group and friends for stimulating scientific. Any questions?
Thank you very much, Raphael. Very, very interesting. So I'm just going to wait for some questions to come in through the chat function. Just as we're waiting, maybe I could ask you, your plot through the Barra Velha formation and showing the increased peaks in phosphorus towards the very top of it, just before the salt, the body.
Yes.
But I wasn't quite sure. Is it the yellow squares? Is that the shrubby facies? Because it looks, if so, it looks like there's lots of shrubs below those points when you have low phosphorus.
Yeah, there are definitely shrubby facies distributed throughout the core, but apparently there is a higher occurrence of these shrubby facies here, because here we have them also intercalated with, for instance, more spherulitic facies throughout the whole section as well.
It would be nice to see a cross plot, wouldn't it, of that directly?
Oh yes.
Great. So we've got some questions coming in, Raphael. First of all, from Ramon Mercedes Marta. Great talk. how similar is your concentration of calcium or over alkalinity experimental values to those in recent alkaline lakes?
How similar the concentrations of which elements again?
Calcium concentration over alkalinity.
Aha, okay. Yeah, that's a good point. So the concentrations of, for instance, calcium, so far I have worked with essentially kept constant the concentration in the range of 10 millimoles. So not lower, not higher than that. Alkalinity, well, I can say that TAC, I have worked with 10, which is somewhat high. So, but I have not changed it, and I have not had time to discuss about this as well. But so far, I just tested a few, changing few parameters like phosphate content, but trying to keep anything else as constant through experiments.
Okay, thank you. Ed follows asked a very similar question to mine about the correspondence with the high P spikes in the top 70 meters corresponding to particular lithologies. And then Ramon asks again, if they are too different, could you explain why?
Too different? What is it exactly?
I think it's just following on from his previous question. I think you probably
all right. Too different, you mean in terms of the testing, the parameters using in the experiments and those observed in modern lake environments?
Yes.
No, I wouldn't say it's too different from alkaline lakes, although we know that some of the alkaline lakes that are not exactly two that are the same. They vary, certainly. So I wouldn't say that these conditions are entirely different from an average alkaline lake.
And probably just the final question we have time for from Ben Bio. The Cretaceous is quite hot, so is the climate variation, does climate variation impact phosphorus concentrations? So I guess it's a question about weathering rates.
Yeah, that's a good one. Yes, I think it might have influenced indeed, as we had a rifting environment, and we may have had catchment from watersheds and also groundwater inflow. So yes, definitely.
Great. Well, thank you very much, Raphael. As I said, we can continue discussions on a dedicated forum and Seds Online, which will be, this is obviously a really fascinating topic. So thank you very much, Raphael. So we're now on to our next speaker, Diana. I'm just going to hand over straight to you, Diana. So thank you very much.
Thank you, Rachel, and thanks everyone for tuning in. My name is Diana Sahy. I'm a geochronologist with the British Geological Survey. I noticed that generally the talks in this session were about proper limestone successions, and really old rocks. This is going to be slightly different. So the topic is, I think I shared the wrong screen. Let's try that again. Nope.
Diana, that looks fine. We can see it.
Oh, okay. For some reason, it shows up on the wrong screen for me. All right. So the topic is the chronology of past seafloor methane emissions along the US Atlantic margin. And in order to be able to talk about that sort of thing, we need archives of those past methane emissions. And these are available in the form of so-called seep carbonates or methane-derived autogenic carbonates, which form at cold seeps. These are places where methane-rich fluids are vented into the ocean. And we need a dating method, and so we tend to analyze these rocks by means of uranium-thorium geochronology. What's the point of doing this? Well, there are a number of implications. So basically what we are trying to do is look at triggers and governing mechanisms of regional scale subsurface fluid flow. And that is of interest to the petroleum industry and also potential applications along the lines of carbon capture, storage, potential geothermal energy use. There are paleoclimate and more likely paleoecological implications. For my own work, this relates mostly to ocean acidification rather than any input of methane into the atmosphere. And there is a geohazard aspect to it as well, because a significant proportion of the methane that is stored in seafloor sediments is stored as a solid, as methane hydrate, and hydrates are stable under specific low temperature and high pressure conditions. And the concern is that if a large quantity of hydrates were to dissociate relatively rapidly, then this would lead to widespread slope failure, submarine landslides, and so that's a concern in terms of hazards. I should clarify, however, that the sort of timescales I work on are still very much geological, so we are looking at tens of thousands of years, glacial interglacial cycles, rather than the sort of 1, 10, 100-year timescales that are more societal relevance. So our study area is a roughly 1,000 kilometer stretch of the US Atlantic margin from New England in the north to North Carolina in the south. And the discovery of methane seeps in this area is relatively recent. The first paper was published in 2014, but since then, our colleagues from the USGS have inventoried something in the region of 800 active methane seeps on the margin at the range of depths. The upper limit of the gas hydrate stability zone along the margin is currently at around 500 meters below sea level. And because of this, we tend to classify the seeps in terms of their position relative to that boundary. So in this talk, we focus on shallow seeps. These are situated above the top of the gas hydrate stability zone at depths of 300 to 400 meters below sea level. And we also look at deep seeps. These are well within the hydrate stability zone itself. And in this particular study, they tend to be at depths greater than about 1,000 meters. So our USGS colleagues collected 18 samples along the margin, and those are the basis of the data set I'm going to talk about in just a bit. But first, I'd like to go through how these materials form and some of the difficulties we've encountered in terms of working with them. So the basic idea is that in the vicinity of methane seeps, the anaerobic oxidation of methane creates conditions that are conducive to calcium carbonate precipitation. So you start off with these millimeter scale nodules that are basically carbonate cemented seafloor dirt. And then over time, those nodules grow, they coalesce, they form crusts. This is what we usually refer to as early stage carbonate cement. And then if seepage continues over time, any voids within those crusts are going to become lined with relatively clean aragonite cement. And this is what we usually call late stage carbonates. This mode of formation raises a couple of issues. First of all, although we can roughly differentiate between different generations of carbonate visually, there is no clear stratigraphy to most methane-derived carbonate crusts. So, for example, the top isn't necessarily younger than the bottom. And also, traditionally, only the very clean late-stage materials were seen as suitable targets for geochronology. But the problem with that is that we'd want to analyze different components of our samples. For example, we might want to try and find the oldest materials available because those would most closely approximate the onset of methane emissions in a given region. So we've had to do quite a bit of development work in order to enable us to date the materials that would best suited to answer our questions rather than just the things that were easiest to work with. The biggest problem is that because these carbonates tend to have a lot of impurities, we have to apply quite significant corrections in terms of accounting for the uranium and thorium contribution of any sediment that's incorporated within our crusts. And the impact of those corrections is increases for the dirtiest samples. So once we've done all the math, we kind of have to try and figure out if we actually got the right numbers. So there's a quality control aspect to data interpretation, and there are two tests we can apply in this regard. The first one has to do with the fact that when you calculate the uranium-thorium age, one of the output parameters is the isotopic composition of the fluid from which the sample precipitated. And experience and indeed the literature tells us that in the overwhelming majority of cases for methane-derived carbonates, this is going to be open ocean water, mean ocean water. So the diagram on the left shows our data from the margin. These are the purple ellipses, and they are separated out depending on which seep field they come from. And what you should be able to see, but I can't see on my screen, behind each of these clusters, there's a gray band, which is a theoretical path along which the composition of a sample would evolve if that sample precipitated from open ocean water. And so all the data we've accepted along this particular margin in this particular data set meet that criterion. The second thing we test for is whether the distribution of our dates matches what we actually expect to see based on sample morphology. So these are three samples from a seed field at Baltimore Canyon, about 400 meters water depth. And all three samples were collected within an area of about half a square kilometer. And if I quickly walk you through the results, what we basically see is we get either statistically equivalent or fairly closely spaced clusters of dates from the late stage carbonates in each sample. And in the cases where we were able to date early stage material as well, we get dates that are still reasonable. They still meet that initial isotopic composition criterion from the previous slide, but they are quite a bit older than the late stage material from the same sample, which is what we'd expect to see based on the morphology. But this also raises two issues that we see again and again in ANDAC, in methane-derived carbonates. And the first of those is that it would appear that individual seeps can be active for hundreds or even thousands of years, although this activity is likely to be highly intermittent because we know from observing modern seeps that seepage activity responds to things like tidal cycles and seasonal temperature variations as well. The second thing is that age distribution, even from samples collected within a relatively small geographic area, can vary quite a bit. And this makes it very difficult to take data from a single sample and extrapolate it in a manner that would allow us to draw any regional conclusions, which is why for this type of work, you need fairly extensive data sets. This slide shows all the data we've gathered so far. These are sort of separated out based on whether the samples are from shallow or deep seeps. So shallow seeps are plotted in yellows and reds and oranges. And deep samples are plotted in shades of blue and green. And similarly, the histograms in the back show the distribution of dates from deep samples in blue and from shallow samples in orange. Now, the challenge when your study area is the size of a continental margin is that you need enough spatial resolution to be able to say something about individual seed fields, but you also need enough spatial coverage to be able to say something about the margin as a whole. And in all fairness, 18 samples along 1,000 kilometers of margin is probably not enough to do either one of those things well. It is, however, enough that we can start speculating about possible scenarios. So if we look at the shallow seeps, activity seems to really pick up post last glacial maximum, especially around 16,000 years ago. The only seeps that are older than that are our northernmost locality of the New England margin, which are plotted in yellow near the top. In that particular case, it's likely that glacial tectonics had something to do with seep activity. Further south, that seems implausible, but we know that there is a general warming trend in terms of bottom waters. And we also know that these seeps are above the top of the gas hydrate stability zone, so the methane being emitted is unlikely to come from any significant hydrate dissociation. But what we think might have happened is that increasing bottom water temperatures since 16,000 years ago have perhaps accelerated methane formation within the sediments. And then there's also a chance that any pressure from increasing sea levels serve to force the methane out of the sediments. The deep seeps are also quite interesting. They seem to start around 10,000 years ago, and then activity really picks up about 5,000 years ago. Now, these seeps are within the hydrate stability zone. They are therefore unlikely to be associated with significant hydrate dissociation. They are all, however, connected to erosional features. So,
1 minute to go.
Thank you. So we suspect that the trigger there is the unroofing of gas charged strata through erosional processes. But it's important to remember that our data set is somewhat biased. We are focused on active seeps, and we are focused on materials that are exposed on the sea floor. And so there is a lot more that we can do to improve spatial coverage and resolution and access materials that we were unable to date before. Whatever few seconds I have left, I would like to use for a bit of a public service announcement. My lab at the BGS is part of the National Environmental Isotope Facility. We are funded by NERC. The purpose of the funding is to enable UK researchers to access isotope facilities. We accept applications from any eligible UK researchers twice a year. We particularly welcome applications from PIs who have graduate students, in which case we also provide training for said graduate students. So have a look at our new website, isotopesuk.org. Have a look at the list of things we do. If there's something you like, please get in touch. If there's something you would like, but it's not on the list, get in touch anyway, and we might just be able to make it happen. Thank you.
Wonderful. Thank you very much, Diana, for that very interesting talk. Beautiful sunset. So again, we're just waiting for questions to come in on the online chat. As we're waiting, maybe I could just ask a bit more about the detailed petrography of these crusts.
Well, what do you mean specifically?
Well, just could you say a bit more about anything about the crystal habits.
So we've done a small amount of thin section work, and we've done a small amount of scanning electron microscope work. And generally what you see in the early stage materials is microcrystalline calcite. And then the late stage materials tend to be acicular aragonite, just collections, bunches of needles.
Okay, so do they okay, that's great. I'm just got my eye on the chat here, waiting for it to come through. Nothing at this point. Yeah, that was that was a fascinating database that you presented. I mean, are you planning to go to other areas when we're able to? Is this something that you want to expand upon in due course?
So in the past, we've done a lot of work with the Norwegian Geological Survey off the coast of Norway, and that's already published. And then last year, I was fortunate enough to take part in a Schmidt Ocean Institute cruise on the Pacific margin, the US Pacific margin. And so that's going to be the next site where we are going to work. But the samples are already in hand, so we are just chomping at the bit to get back in the labs. Yeah,
absolutely, as we all are. There's several questions have come in now, Diana. So first of all, from Fiona Whitaker, do you see an expression of erosion and unroofing in seafloor topography around your deeper seeps?
We do. So these seeps are located generally at the bottom or on the flanks of submarine canyons, or they are associated with topographic heights. So in all of those cases, it is quite clear that it's an area of sediment erosion rather than accumulation. Because that was our concern at first when we first saw that separation between the shallow and the deep seeps, was that, okay, the deep sea is sort of a place of sediment accumulation. And so are we only seeing young samples because everything else is simply buried under tons of material? But that does not appear to be the case.
Thank you. And then Kathy asks, thank you, Diana, for an interesting talk. Do you know anything about the temperature of the seeps?
Well, beyond the fact that they are cold seeps, as the name indicates, no, not really. We've been fortunate to work with a fairly interdisciplinary team, so we always have biologists and people who work on the fluids that come through the seas, but I don't think we've carried out any actual temperature measurements. And then any temperature information we get from stable isotopes, we've done a bunch of stable oxygen work on these samples, but it's not clear if the data we are seeing relates to temperature of the seeps or the input of fluids with some weird chemistry coming up from below. So we can't really interpret it in those terms.
Thank you. So Gadella asks, in deeper parts of the continental margin, is there any influence from black smokers, in other words, hydrothermal vents for methane sources?
Not in the areas we've worked in, as far as I know.
Okay, thanks. And then Fiona just wants to follow on. Also interesting is that you mentioned that you see tidal and other influences in the deep systems. Could you comment on that further, please?
So there are two possible scenarios here. As far as I know, there was a paper published on methane seeps off the coast of Svalbard, where they first pinpointed this tidal influence, and it was surprising because the seeps were at depths in excess of 1 kilometer. And because there is this tendency to try and relate everything to hydrate dissociation, they hypothesized that they would see seepage during times of low tide because of the drop in pressure, which would cause hydrate to dissociate. But that seems a bit tenuous when you're under more than 1 kilometer of water. However, on the cruise I mentioned, last year's cruise on the Pacific margin, we witnessed the exact opposite, with seeps being more active during high tide. And so it would seem more likely that it's actually the increased pressure from the higher water column that it's squeezing methane out of the sediments.
Thank you. There's two more questions. We may only have time for one, which is from Pankaj. Thank you for your talk. I might have missed it, but what is the cause for more seeps during the Holocene as compared to the last glacial maxima? Did you have samples from LIG seeps, if there are any? Do you have any comments?
So this goes back to the last bit I said, which is that there is a sampling bias in the data set. In that we only have materials from currently active seeps and materials that are exposed on the sea floor. There have been a couple of studies, not on the Atlantic margin, but off Svalbard and in the South China Sea, where people have actually managed to drill cores through the sediment column, and they recovered seeps, seep carbonates. And those are quite interesting to look at.
Great. Thank you very much, Diana. We have got some other questions, but we're going to have to stop now. But obviously, it's generated a lot of interest. So thank you very much indeed for your presentation.
Thank you, Rachel.
So we're now going to pass on to Marta from Imperial. So over to you, Marta.
Thank you. Can you see the presentation?
Yes.
Yeah, perfect, thank you. So good morning, everybody. My name is Marta Marchegiano, and I'm a postdoctoral researcher at Imperial College. The aim of the study I'm going to present to you today is to reconstruct variation in the hydrological cycles and temperature in the Hampshire Basin at the end of the Middle Eocene Climatic Optimum and its recovery using a multi-proxy approach, mainly the analysis of ostracoid assemblages and carbonate clumped-isotope. Okay, so the study of global warming event, it's important to understand how the Earth responds to this event and thus to create model for future climate impact. In this framework, MECO, it's an outstanding event because it's a global warming anomaly where an increase up to 6 degrees of the seawater temperature have been recorded in several sites. These events happened around 40 million years ago and has a length of around 500,000 years. The main feature is a negative signal of the delta 18, almost recognized everywhere. However, concerning the delta C13, it was very variable in terms of timing and amplitude. So Hampshire Basin is located in southern England, It is the most northwesterly part of the Anglo-Paris-Belgian basin. It is an asymmetrical elongated syncline where the Paleogene sediments were deposited during a series of marine regressive cycles. And it is one of the best preserved Paleogene sequence worldwide. So this is the reason why we decided to analyze the Barton Clay Formation, the section of Barton on the Sea, that is located here, and that include MECO event. Okay, so the almost 44-meter section have been lithologically described and sampled every 1 meter, more or less, by Cedric Jones and Annabel Dale. The bottom of the section is coarser and became finer going upward and ended with a coarser level of very fine sands. So for all the 34 samples have been analyzed, the ostracods fauna have been analyzed. Ostracods are thin aquatic crustaceans that can live in different kind of aquatic environment. They present a low magnesium calcitic shells, and they are very sensible to several ecological parameters. For this reason, they are very useful in paleoenvironmental and paleoclimatic reconstruction. However, because of their very small size, it's still quite challenging to use them for carbonate clumped-isotope. And for this reason, we used gastropods from 13 samples. As the pictures show, they are very well preserved. And after FTIR spectroscopy analysis, we knew that they were pure aragonitic shells. And this is quite uncommon to find so well preserved fossils in Eocene sequence. So I would like to spend a few words about the carbonate clumped isotope technique. This is still quite a new technique, and to my knowledge, is the first time that has been applied to investigate MECO event. It is based on temperature-dependent abundance of C13 and O18 bonds in CO2. That is higher in lower temperature, and is quantified as CAP47. This abundance is directly correlated to the temperature of precipitation of the carbonate. But what are the main advantages of this technique compared to bulk isotope, for example? Firstly, they are not sensitive to delta 18 of the water, as you can see from the formula. And this is very important in coastal environment where we can have a riverine input. Then once you have the temperature, you can directly calculate the delta 18 of the water with this formula. And so this allows us to reconstruct change in the hydrological cycles. Moreover, the vital effect is absent for several organisms, including gastropods. So a total of 20 species belong to 17 genera of ostracods have been recognized. Those species are, most of them are endemic and extinct, so it's hard to know their ecological preference. However, compare those analyses with geochemical ones allowed us to understand better our paleoenvironment. The ostracod data have been analyzed using a cluster analysis, and 4 different ostracod assemblages plus a barren event have been recorded. Then we used the results of ostracod assemblages plus the delta 18 calculated in our section to constrain the chronology. And so we compare our data with those obtained by Dauber et al. In 2011 in the Barton Clay Formation at Halong Bay, that is just in front of our section. And we are quite confident in saying that our section includes the end of the MECO event and its recovery. So then I plotted the cluster analysis results again the stratigraphy to reconstruct the to see the different paleoenvironment along the section. Starting from the bottom, we have the first interval that is just made from 2 samples, so we can't recognize the paleoenvironment. However, it's important to notice that this association is different from the others in the upper section. And moreover, we can notice the presence of ostracods at the bottom of the section that then totally disappeared going upward in the interval B. So what could be the reason why we don't have ostracod in this interval? Firstly, the presence of an environment that is not suitable for ostracods. And what could be, if we look at the lithological the lithology, we saw that this was the coarser section. So maybe we can hypothesize the presence of a shallow environment with high energy and so not suitable for ostracods. The other hypothesis could be a no preservation of those shells. However, the good preservation of gastropods, as well as the total absence of ostracods, suggests that the first hypothesis could be the most plausible. So we interpret this environment as a shallow littoral. Going upward, we use the abundance, the species index, equitability, and all the characteristics of all the species we recorded to interpret our paleoenvironment. So the interval C was a shallow epineritic, then we have an epineritic infraneritic, and then going upward the littoral shallow epineritic. In the last interval, ostracod disappeared again, so we can imagine an environment close to that one that we had in interval B. So ostracods indicate a deeper followed by a shallower trend. Analyzing the geochemical results, we have, starting from the left, the temperature recovered by clumped-isotope. Then we calculate the delta within of the water and the bulk isotope, the oxygen and the carbon. So it's important to notice that even if it's not exactly the same, the trend pointed out by the temperature from clumped isotope is the same that we obtained from bulk oxygen isotope. So the interval B, as we said, was a shallow littoral. And looking at the temperature, we have higher, warmer temperature, so indicated from both clumped and oxygen, delta 18. So we can say warm temperature. Then looking at the delta 18, we have a clear freshwater input, and the delta C13, also a negative delta C13, indicate an input from the continent. So we interpret this one as a warm, humid event. And this was the MECO. So going upward, we are in a shallow epineritic environment. The trend of the temperature change through colder temperature. And we still have a freshwater input and negative delta C13, so called humid period. And this was the end of the MECO. Then the infraneritic, the epineritic infraneritic environment, we recorded a change in the trend. So we are again through warmer temperature. And we still have a freshwater input, but the trend is changing in this case, as well as for the delta C13. So we are in a warm, but still humid temperature event. And this was the recovery after the MECO. So the last interval, we had a great increase in temperature. However, in this case, the delta 18 of the water indicate a more marine signal, and you have a more positive delta C13. So in this case, we interpreted this interval as a warm but arid because we don't have more input from the continent. So to conclude, the ostracod assemblages recorded a change from shallow to deep to shallow marine environment in the Hampshire Basin during the Middle Eocene. These mirror the clumped and bulk isotope signal that point out changes in temperature and input of continental freshwater into the basin. And in general, this multiproxy approach has a tremendous potential in reconstructing paleoclimate and continental margins. Thank you.
Thank you very much, Marta. Great. So I'll just wait for the questions to come through on the chat function. perhaps I could ask a question while waiting. I think you presented your species data as a percentage of the overall population. I might have seen that. But do you have any data on changing abundance as well? In other words, do we know that there are times when you have effectively ostracod blooms?
Yeah, exactly. If you look at this diagram, the first, starting from the left, is the abundance. So it's the real number of valves that I found in each samples. And for most of them, I was close to 300. That is the number required to have a good statistic representation of the assemblages. But in this interval, the interval D, the number of ostracods decreased enormously. So I found very few valves. And yeah, so this could have an importance. But for the moment, I don't know exactly what could be the reason why there is. It could be maybe a drastic change in and some parameters in the environment. I exclude a big change in the bathymetry because normally the ostracods presents the assemblages doesn't change a lot. What changes is the percentage, the abundance of certain species. So I'm not expecting a big change in deep change.
So we don't have any blooms of, very low diversity or single species blooms. That's really what I was getting at.
Yeah, this one, for example, is the Leguminocyte striato puntata. Is the most abundant species. Yeah. And yeah, it's particularly abundant here. It's true that if we look at the percentage, I mean, the big abundance is also because the others are not really present. But for example, here I had around 100, less than 100 species in this part, but here in the upper part is close to 200 samples, 300. So it's statistically, I mean, it's statistically important. We have a real abundance of these species compared to the others.
Absolutely, great. Thank you very much. We've got lots of questions coming through. So from Hossein, great illustrations, Marta. She's wondering if you've performed any clay mineralogy, especially within the finer upper part of the section. And if so, follow the warm cold results. Thank you.
Unfortunately, no. I'm just working on the ostracod assemblages, and then I use the gastropods for the clumped analysis, because the main aim of my postdoc was to learn this new technique. So I wasn't focused on that. However, I would like to, because I just work on the samples, so but I would like to go back to sampling more this section. Unfortunately, this is not a good period, but maybe soon.
Thank you. And then Anna asks, nice and clear talk. Why ostracod assemblages? And can this method be applied to other fauna or microfauna?
Sorry, I didn't understand. Why did you choose ostracods
for this type of analysis? And could the same methods be applied to other fauna, for example, or microfauna?
You mean for the environmental interpretation or for the clump?
Well, I think just the whole methodology.
Okay.
Why did you choose ostracods, and then could you apply it to another set of?
Okay, the only two fossils I found, because normally macrofossils doesn't have the same resolution of microfossils. And so concerning microfossils, I just found ostracods and forams. And I did my PhD on ostracods, so I'm expert in ostracods. This is why I choose more ostracods. And yeah, and then because in those sections, I found some forams, but were bentonic foram. So all the fauna presents were bentonic, and I didn't find any planktonic forum in my samples. So I think, I'm not expert on forums, but I think that we could have the same information when we are speaking between ostracods and forum in this kind of environment. But if someone wants to study the forums, I'm happy.
Thank you. And then Hugo's asked, thanks for the talk, Marta. Maybe I missed it, but do you observe any changes in ostracod species with chemical variations in chemical conditions?
You mean in the preservation of the shells?
I think you probably mean seawater chemistry conditions, I'm guessing, but it just says with chemical conditions variation.
The problem with those, normally, for example, during my PhD, I was working on quaternary ostracods, and with those, at the time, it's easier to know information about the, about the water and salinity and everything. However, with those species that are extinct today, it's hard to understand something so specific related to the chemistry of the water. This is more thanks to geochemical analysis that, for example, I reconstruct the delta weighting of the water to understand if there were some freshwater input. But unfortunately, in the part where I expected to have more freshwater input, I didn't have ostracods. It was here, so I didn't find. I didn't have any information about different chemistry from ostracods at the state. I'm still working on the data. Actually, this was the first results I had, so I still have to put all my results in a more general framework to understand better what's happening. Because these are the results from the regional. What's happening in the Hampshire Basin, but I would like to put this in the more general framework.
Thank you very much, Marta. We've got some other questions about size changes and preservational changes, but I think they're going to have to wait for the, because we need to move on to the next presenter. But great questions there in the online chat, and I hope that you'll have a chance to look at those and discuss them with Philip and Anna and Elias. So thank you very much, Marta, for a great presentation. So we're going to move now to the final presentation of this session, given by Nick Roberts. And Nick is presenting a poster. And this has a slightly different format. We ask you please to go to the poster that's uploaded in the Seds Online. And I don't know whether, Nick, you're also going to share your screen with us, which has a poster and there we go, and talk us through it. So this is a much shorter presentation. The posters are only 5 minutes. So I'm going to hand over to you, Nick, and maybe you could give us 2 or 3 minutes, talk through your poster, and then we could have a couple of minutes discussion. So
okay, can you hear me?
Yep, that's fine.
Okay, so I'm Nick Roberts. I'm from the geochronology and tracers lab, the same lab that Diana Sahy was from talking about a moment ago. They're based at the British Geological Survey. And again, another plug that we're part of the NERC facilities, the National Environmental Isotope Facility. And if you're interested in these methods, then please do get in touch with us. So just to give you a little overview of the poster, let's start at the beginning. I've got a little bit of the brief history, just some timeline. So the traditional technique of dating carbonates was dissolving them up and doing chemistry on them. And that started in the late 80s. There's been a handful of publications, or well, a few handfuls on different settings, but it never really took off as a popular method due to its limitations. In the late 90s, David Richards at Bristol managed to date some quaternary material, and that's really where the method has remained, is within dating speleothems. We started work using laser ablation in around 2009, 2010. We published the first paper in 2014, and we published the reference material, which is critical for the laser ablation work, in 2017. And there's been a handful of reviews, some of them listed here. So yeah, we're talking about the laser ablation method. We can do this on thin sections or polished blocks or chips mounted in epoxy, and we normalize to a carbonate reference material. And various labs are now trying to find better materials or materials with different matrices, such as dolomite. This is just, this plot over here, just an example of some of the petrography. As with all geochronology, you need good petrography to get good dates and context for your dates. These boxes over here discuss a few of the limitations. So one of them that's really the biggest problem for carbonates is the fact that often the uranium is very low and the initial or common load is very high. And these density plots just show compilations of some of the material I've worked on and the variable uranium within those. Another limitation for dating young carbonates is disequilibrium. I won't go into any detail in that, but if you want to date things that are below, say, 10 million years, then understanding disequilibrium in the fluids that grow or precipitate your calcites is something to pay attention to and affects the accuracy of your ages. Since 2014, various labs have now taken up this method, and we've been applying it to various things, just silicon fibers and fracture filling veins, breccia cements. There's been successful dates of paleosols and caliche, some limited work on diagenetic cements, but that's a growing field, speleothems, and people have now started working on sediments and orthogenic limestones. In terms of applications, I've just put a few examples on the sort of work I've been working on. So I've mostly focused on things like fractures and faults, and less so on things like actual sediments. At the top one is some nice exposures at Cedric Bay, which is quite a famous locality along the Yorkshire coastline. And we've managed to date some of the cements that fall within those fractures, and they're around 60 to 55 million years. These plots here, these are backscattered element images, just to show that within actual single crystals, you can get multiple dates. So these grew over quite a protracted time period within fractures within the crust. This third example down here is just to show that with this combination of in situ methods, we can do things like in situ ion probe carbon and oxygen isotopes on some crystals. We can take these same crystals and then try and date them. I've been involved in studies looking at hydrocarbon charge. This first one is looking at basement fractures, where sediments in calcite has propped open fractures deep within the crust, and that's helped hydrocarbon migration within this place. This is the Rona Ridge offshore. And this study at the bottom here, this is Nathan's work that he talked about yesterday, these microbialites. So we've actually managed to date some of the shrubby and spherulitic type calcite and dolomite, and got some pretty nice age data. And then finally over here, this third application box, just showing the range of ages. So uranium lead, we can go down to, we can cross over uranium thorium, which tops out at about 500 to 600,000 years. This is a speleothem dated at 1 million years. These are some ages, you can't really see these. These are about 500 billion years, pretty imprecise, but they do overlap the known ages of deposition. And I was pretty skeptical about working on even older material, but this is North China Craton platform carbonates, which host some of the early fossils. And we've got a pretty good date here of 1 billion years with a pretty good uncertainty of 30 million years, which is pretty good for this method.
Great. Unfortunately, the screen share is about to be cut, but really fascinating stuff. Very much indeed. There's unfortunately no time for questions, but there again going to be any questions will go into the breakout rooms, and there will certainly be many. So thank you very much indeed. A great, powerful technique. So just before I hand over to Kathy, I just want to thank all the speakers in the last session. If we needed any testament to the vibrant community of interdisciplinary work we have in carbonates, this was it. So thank you all very much for your terrific presentations, and also for so many fascinating questions coming in. Thank you all. Over to Kathy.
Thank you, Rachel.